I spent about 20 years working in ocean, and Bruce got into and worked on a couple of ion channels. Burlington. Prior to that, Harvard in the lab. Any peer review. So without further ado.
How about now? Okay, good. I think I turned it off rather than on. Okay, so what people in industry call what Dan and I are going to talk about today is lead identification. That is trying to find a small molecule. We're talking about small molecules here, first of all, and try to find a small molecule lead that can then be chemically optimized to give you a drug. And we're going to talk about two different ways of doing lead identification. Oh yeah, so there we go. So just to sort of orient you where in the process we are, we're very early. And there are a number of ways you can do lead identification, and we're going to just talk about two of those, but just to notice, you know, the usual thing you hear about here, people find stuff in the literature. You can also modify substrates or transition, go for a transition state and make a transition state analog. And then what people often do when they're just screening the drug library, trying to repurpose things, a subset screen. So I'm going to first talk about high throughput screening. And how do we screen in high throughput? This is actually, you know, we don't have one of these here, first of all. This is the industry view when you're screening 1.2 million compounds. Here, we screen 10-fold less than that. So we have little baby versions of this. But the idea is the same. We use 384 well plates. We do assays that, for the most part, are mix and measure. We don't like washing. We don't like having to take things out and, say, run a gel. As I said, laboratory automation. The other thing that's very important is because of the cost and the large number of compounds, typically we screen one well. So that's a single concentration, one time to find a compound. And that has certain downstream implications. On every plate, we have control wells. We have negative control wells, positive control wells, and typically to try to look at and keep track of the sensitivity of the assay, IC50 control wells. Here you can see they actually sort of missed the IC50 a little bit, but this is the basic idea. And then there's this parameter that screeners use to talk about how good an assay is. And that parameter is called the Z prime factor, not to be confused with the Z score, which is mean, correct, you know, normalized mean, so the mean 0 and 1 is one standard deviation. This is a Z prime factor. And the way it's calculated, you can think about it as starting at the mean up here, come down 3 standard deviations, start at the mean down here, go up 3 standard deviations, And we're looking at the relative size of that window relative to the difference between the two means. And it's a dimensionless number. It goes from 1, which is considered a perfect assay, to negative infinity. 0 and below are considered to be useless. And anything above about 0.5 is considered to be good, although higher is better, because since you're only screening at a single concentration, the less noise, the more power you have to find actives, particularly weak actives. So, but give you some feeling for how this relates to maybe things you've done in the lab. This is a dose response curve. By the way, so this is a Z prime factor of minus 0.5, useless for HDS screening. But as you can see in the lab, you're doing single compound and you're doing a concentration response. It's ugly looking, but you can fit it and you actually get a pretty reasonable idea of how potent the compound is. For single point screening, useless. Look at all these things up here that are going to look like they're active, just random. This is all inactive. This is just random noise. Conversely, the threshold of a good assay, Z prime factor of 0.5, now you get, of course, a beautiful dose response curve fit, and now the data is much more nicely distributed around 0, and so you have a much better chance to see weak hits up in here. So other than the Z prime factor, what makes a good HDS assay? And, Steve here, your favorite one. First and foremost, biologically relevant. And, given the choice between slower, more expensive, but biologically relevant, and faster, I would strongly encourage you to go for that. I have a hard time getting people to do that, but that's size of the assay window, precision, which is really what Z prime factor measures, long-term reproducibility. You have a standard compound. If you run it now, if you run it a month from now, both in dose response, do you get the same result? Let's see, sensitivity to inhibitors, which we'll talk about in a minute. And I think most of these other things we're going to talk about later. Just mention detergent. If it's a purified protein assay, detergent below the CMC is critical. Otherwise, you get these high stoichiometry binders that are really, can fool you. I think you're going to talk about those. Dan will talk about those. So, but a reminder that not just in fragment screening, but in HDS, detergent is critical. Reagents being at least somewhat stable over time. Since we're talking about lower throughput robotics here, on the order of hours at least. So if your reagent dies in 20 minutes, or your protein after it's thawed out and diluted, you're going to have a very unpleasant experience. And then, of course, since you're going to be doing a lot of this, ease of use and minimal number of steps, and again, 130,000 wells, actually more than that, 130,000 compounds, about 160 to 200,000, depending on how things go, wells at the end, cost. Oh, lovely. Okay. All right. So assay technologies. There's typically more than one that will work. While I remember, there are backup slides to this, and the Spark people have the PDF of this, and there's a whole bunch of slides on different assay technologies that are enabled at Stanford at the HDBC. I am not going to go through those, but they're there for you to look at if you'd like. Okay, so yeah, all the kinds of things you have to think about. This is dealt with in those slides. This you either need to know or you need to come talk to me or to David Solo-Cordero, and we can give you an idea of sort of what you're going to be in for. And I will talk some about this at the end, and then assay-dependent ones, and then Dan will talk about chemistry-dependent false positives. False positives come up a lot in this kind of discussion. And, just not to belabor the point, but often when developing assays for HTS, there are trade-offs. There are trade-offs between how good your assay is, measured by Z prime factor, how sensitive it is to inhibitors. You can make an assay that has a terrific Z prime factor by making it very insensitive to inhibitors, for instance, in enzyme assays, and cost. And those all have to get balanced. So I'm going to cover both cell-based and purified enzyme assays, but I'm going to start with the latter and talk about substrate concentration first. So often, and we're going to try, people here do activator assays, but I'm not going to talk much about those. Those tend to be a bit more idiosyncratic. I'm going to talk about inhibitors. And when you're looking for inhibitors, often people don't know what mechanism they necessarily want. And so if you're willing to take any of those three, then your best place to be is to have a substrate concentration equal to the Km, because that gives you the best sort of balanced sensitivity for uncompetitive, competitive, and non-competitive, of course, doesn't care. Let's see. So the other thing is that, people are trained, you go to take biochemistry, enzymology, typically they tell you you don't want to turn over more than 10% of the substrate. And it's true. If you want really good quantitative data, that's what you should do. But we have to, in HTS, we have to cheat. Again, we're balancing various things. Usually you get a better Z prime factor if you turn over more of the substrate. And get an idea of what's going on here. If you look like out here at the end, the things that have really sort of depleted their substrate, you can see there's less sensitivity to inhibitors out in that range. And that's been plotted here. This is the IC50 for the compound at very low substrate turnover compared to what you get if you go much higher conversion. And it turns out you can get away with a lot. You can get away with, converting 50, 60% of the substrate with a less than twofold loss in sensitivity. And that's going to give you a much better Z prime factor. And in fact, most HTS assays, unless they already have a huge window, are run maybe at 50% conversion or thereabouts. Okay, so things, you developed an assay, one of the things you need to do is to characterize it. Again, we need a very robust assay for HTS. So obviously one of the things you want is linear increase in initial rates with more protein. It's kind of frightening that you sometimes don't see that. I talked about that you don't have to be absolutely linear with time, but this example is, and in fact shows you that when you run the assay at 0 hours or after the reagents have been on the deck for 21 hours, you get the same result. Many of the assays run at Stanford don't have prototypical compounds, so then you can't do this. But if you do have compounds from the literature that hit your target, maybe aren't drug-like, which is why you're looking for new things, you would like to see a very good correlation between PKI in your assay that you've just developed and the one that is considered the absolute definitive assay. Maybe it's an HPLC assay where you measure both substrate and product, for instance. And finally, the libraries aren't sitting there in the plates as powder. They're sitting there dissolved in DMSO. And one of the things you need to know is how sensitive the assay is to DMSO. In this particular example, even up to 10%, everything was good. Not always what you see. Sometimes it'll start to do that. The other thing is that DMSO actually enhances the solubility of compounds, so that in fact, if the assay can put up with like 5%, you may want to run it that way. The fragment screening is almost always run that way. And the reason is compounds are much more soluble in 5% DMSO than they are in, say, pure buffer or very low DMSO. All right, let's talk a little bit about cell-based assays. And here I'm going to talk more about design of assays. I mean, again, when you're going to a cell thing, you're trying to look for a biological model, either phenotypic assay or look at the activity in a pathway. Ideally, differentiated cell line or differentiated iPSCs. Most of the screens that have been run at HTBC are classic reporter gene assays. I'm going to encourage you not to do that. I would encourage you to instead to try to use an assay where you can put a reporter into the native locus, use the full enhancer and control sequences of the cell. And this is one example of how you can do that. So what this system is, and it's designed to be put in with CRISPR-Cas9 because it's a short thing that you're going to add on to a protein, it's a luciferase. It's not firefly luciferase. It's actually a brighter luciferase. But it works much the same as beta-gal. You get beta-gal complementation. In the case of beta-gal, it's a short peptide. Same thing here. You use CRISPR-Cas9, you put it into the one of the native loci in the cell at the C terminus of the protein. And now you have this, and I'm trying to say, 11 amino acid tag. So it's small enough that chances are it's not going to have a huge effect upon the expression. But it lets you monitor expression very easily. Okay, second one of these is actually a screen that was run by one of the Spark projects, Justin Dennis. And it's an imaging-based assay. So one of the things that HTBC has is a high throughput imager. And that then allows you either using fluorescent proteins or antibodies to actually look at changes in expression, changes in number of cells that do various things, changes in shape of cells, I say number of cells, changes in the number of cells. What this is called in the field is high content screening, and that's because you can measure multiple things at once. The system we have can pretty easily measure 3 different fluorophores. One of them you're going to want to use as a thing to find the cell, so it's going to be DAPI, but you can watch two other variables. And then the last one I wanted to talk about, and this actually was also used for a Spark project, Stan Cohen's Spark project, looking at protein-protein interactions in cells. And so this is raneloluciferase that's been split. And only when it has the 2 pieces have low affinity on their own, and only when they're recombined do you get luciferase activity. And then so it was then used to look at the interaction, protein-protein interaction inside the cell. And there were multiple reasons he did it, but one of the reasons is because one of the 2 proteins he was looking at is a very unstable protein. Yeah, actually it's this, but yeah, it's a very unstable protein, and so you couldn't purify it. All right. Can't do all the same things with cell assay. You can do with purified protein assay, but you can still look at stability over time. You have the compounds, you can look at the pharmacology, and you absolutely should look at the DMSO sensitivity. Cells typically don't like more than a few percent DMSO, if that. All right, so one of the other things really encourage you to look at after you develop an assay is to look at what happens just with plates that have nothing active in them. They're just filled with DMSO. And this is an example of, so in that case, if you don't have any spatial biases, you don't have any problems with the assay, you should just have random noise. These three are Q-cube plots, and they let you then look and see how close to random noise you have. The other thing you can look at is if, say, one of your instruments, one of your pipetters, your reader or something, gives a biased result, you're going to get correlation between the two samples, and that's actually what you see here. Well, plate 1 and 2 are correlated, whereas plate 2 and 3, they're not correlated. So for some reason, the way you ran this, you've ended up with the same kind of noise. All right, false positives. Now again, Dan's going to talk about chemistry-based false positives, and there's even a lot more ways you can get that. But things that typically show up are, for instance, if you use a luciferase reporter, luciferase inhibitors. In a lot of assays that use fluorescence, not so much the imaging ones, but if you're reading fluorescence intensity, things that absorb either the exciting light or the emitted light and fluorescent compounds, both can cause problems and need to think carefully about how you're going to deal with that. There are ways of, I should say, there are ways of at least seeing whether those problems show up in many of these assays, but it takes careful thought about that. And then, you know, there'll still be some of that. So one of the things that's important is downstream of the initial assay, when you're trying to validate the hits, orthogonal assay technologies. Use completely different readouts. Counter screens, either, you know, well, I guess like example could be luciferase. You can buy luciferase from Sigma. Measure what your compound does to luciferase. That's a counter screen. There are also a bunch of counter screens that are important for chemical false positives. Are you going to talk about that? I hope. Okay. And finally, for the orthogonal assays, the lower throughput assays with a wash step, which means you treat the protein or you treat the cells, you wash the compound out and you do the readout, then the compound can't interfere with the readout anymore. Do not want to do the HDS that way. You will not enjoy it. This is just kind of an overview of the whole process then. If we kind of back up, you've got to make either purified protein or your cell line, develop the assay. I would strongly encourage you to do a pilot screen, either some little piece of the library along with some DMSO plates. See how well it works before you go whole hog. A primary screen, figure out what the hits are, confirmation screen and counter screens, and those are typically done in dose response. Kind of look at which things are pains, other things like that in silico profiling. Also, you know, we're looking at your hits in PubChem, because all of our compounds, unfortunately, are in PubChem, and see whether how many assays they've been in, how many assays they were active in. You don't like the one that's been in 130 assays and it was active in 35 of them. And then we can cluster them and see if there's any patterns that show up with chemical structure. And many of you may have heard the medicinal chemists are kind enough to meet with people who have run screens and to look at their compounds, and they often find some things that are good and other things that they tell you should stay far, far away from. One of the other things I would encourage, if not before this meeting, certainly after that, when you're reordering compounds, is to confirm that the compound that you get is the structure that you thought you were getting, both by LCMS and NMR, because you would be surprised how many times it's not exactly what you thought, and even in the ugliest cases, it's been run on LCMS and it's a single peak and it's the right molecular weight, and they do the NMR and it doesn't match what it should be. The right mass, but the wrong structure. Hit validation is those secondary assays. And hit expansion is something the chemists do. They help find similar structures for you then to look at. Eventually you need to synthesize things, but often initially you can order things. All right, almost done here. So for phenotypic and pathway screens, cell-based assay, you get done and you don't necessarily know what the molecular target of your compound is. That can be okay, although it can also make life more complicated, both to get more funding and to get people excited about your project. And so there are ways of actually deconvoluting things. There are sort of in silico things. CESTA is a way of thermally denaturing proteins in a lysate or in a cell, and looking then at and being able to figure out what the compound is interacting with using mass spec or antibodies. I mean, there's sort of different flavors of this. You can overexpress proteins in an array, for instance, and see if any overexpression then shifts your dose response. There's the classic things of making a affinity column or photo affinity labeling. You typically have to know something about the SAR of the compound before you can do that. In infectious diseases, they often select for resistance and then sequence and find out what changed that gave you resistance. And then finally, there's this technology developed by, among others, Mike Bassick, who's here in genetics, that, well, uses different versions, either, say, 20x shRNA or CRISPR-Cas9, knocks out genes and looks for synthetic lethality. And you can get growth synergy, although I think it's worked best for synthetic lethality. And there's at least one Spark project that managed to identify the target of its compound very well that way. All right, purified proteins. And these technologies are both good for that, and they're good for fragment screening. SBR, which many of you know I'm a big proponent of, protein-observed NMR, it can be a great technology. It is limited to under about 40,000 molecular weight or so. ITC, not good for a large number of compounds, but if you have something like a protein complex, which is hell for the SBR, and is not, I mean, you're not going to be able to do that either. That's the way to go. Thermal shift looks at the melting of proteins. It doesn't always work. It works best, if you have low affinity compounds at least, it works best for proteins that aren't super stable. So they have a relatively low TM. And, absolutely the gold standard, protein crystallography. You get a crystal structure, you not only know your compound interacts and how it interacts, but you gain a lot of other information. You really give a big head start to the chemists. Finally, sort of for fragment screening, there are a couple of other things available here. One is ligand-observed NMR, which doesn't give you as much confidence that you're interacting with single site on the protein, but is a good screening method. And just mention that one of the faculty members here has a substantial size fragment library that's at much higher concentration and volume than what we have in our fragment library, and for doing crystallography. Direct interaction of compound with a protein? With the target. Yep. Or, I mean, the same things could be used for actually for screening. So fragment library is much smaller. Our fragment library is 5,000 compounds, for instance. So you actually can, many of these things you could screen an entire fragment library, or you could screen your confirmed hits from the HTS to get, and that gives you, if you, for instance, in protein observed NMR, you see it's binding at one site, that's extremely good evidence that that's what the compound does. It has a direct interaction there. I know. I thought that was only SPR. Do they actually have protein? Oh, God. I mean, you know, we've had this discussion here before. The FDA doesn't require that you know the target. The people, the VCs who are giving you money may feel differently about that. Okay, so let's see, I just jumped, no, there we go. You know, they're fairly straightforward things. Best assay that you can develop. It's worth the time and the effort not to rush into screening. Screening is expensive. It's going to use, you know, most of a year's worth of Spark funding. So you're not going to do it multiple times. Worth the effort to really come up with the best assay you can. Confirming actives in orthogonal assays or using counter screens. And, you know, consider what kind of undesirable mechanisms of action can mess up your particular assay. Yeah. Oh, one thing we hadn't talked about, but Dan will mention that, is when you get down to a tractable number of hits, 10, 20, 30, spend some time with SciFinder. SciFinder is a chemistry-based thing like PubMed. Lets you look up all the things that have been published about your compound. Sometimes, if you get lots of publications when you search and they're for a large number of different targets, that's not a good sign. And yes. I have not. So my understanding at the current stage of that agreement is they want things that have already been sort of off the end of that flow chart I just showed, and that there are a number of those sitting around campus waiting for more MedChem, and that's where they are. I do know the Stanford person who comes this fairly often, actually, is Mark Smith. I used to work at, he and I used to work at Roche together. And he does tell me that things are going to move earlier in the process, although Takeda themselves does not want to fund the early stuff. They'll provide some input, I guess. No, it's these guys, Mark Smith. Yeah.
Advisors here that
Yeah.
fill up program.
Yeah. But they're not Takeda, are they? No. No, that's what I thought. That's the question. No, but that's it. Yeah, we do. I mean, there is MedChem input, and yes, unfortunately, they tend to find lots of ugly things. Things that I didn't even know we had the ugly things in our library. Well, it said it would cost to run to do the final stages of developing an assay, to run it, confirm the hits, and order some number of them. Probably the total price tag of that's about $50,000, which is about a year's Spark funding.
Ready to start setting up your screen. What sort of support along the way?
Well, yeah, so both David Solo-Cordero, who runs the HTBC, and I are available to talk to people to discuss their assay with them. For the most part, the actual lab work needs to get done by the investigators.
Another question. I know we have some.
Oh, yeah, but a few hundred. Yeah.
And they're sort of pooled, right?
But when they're not pooled, they didn't get completely purified. So that's a little different, actually.
Have people been using them? Have they been effective?
My understanding is there have only been a few hits and that they haven't been able to figure out what the active thing is.
Question is, again, your faculty postdoc. Have a crystal structure for your postdoc. Do you automatically want to go the fragment-based route? How do you decide which route you're going to take?
I actually then would talk to the medicinal chemist. I think that's the, I mean, if you have a really high resolution, well, if you have a crystal structure that has a compound bound to it, then you might at least think about doing computational-based screening as a first try. I mean, it can work great these days. 10 years ago, when I was in industry, it mostly didn't work so great, but I've heard enough now that it can really work great. The bad thing is, unlike an HTS assay, which, we have how many? 500 plates in the library. After you get through the first 50 plates, I will tell you whether your screen is working well or not, or you should give up. They can't tell you that for the computational one. They basically, until you test the things they predict, they have no idea whether it worked well or not. And so that's the downside. But, other than probably having to order some compounds, it's not that big a deal. Personally, my second place I would go there would be fragment screening then, if you have a crystal structure. Oh. Okay. 130,000 compounds. It's on the HTV website. A lot of it was designed by Kip Guy, who was a medicinal chemist who was at UCSF and then was head of the Department of Chemical Biology at St. Jude's and is now dean somewhere. And, I mean, considering the size, which is small compared to so many farmers used to, had a lot of successes with the library. It doesn't have a lot of redundancy. So when it's clustered, the cluster sizes tend to be 1 or 2, unlike a pharma library where, the biggest cluster in the Roche library had like 500 members in it, using the same parameters, actually using the same tool. It's in 384 well plates, and we don't have a lot of it. We bought it. Oh, it's the same library. So UCSF has more now, but it is the same library that at one point was all UCSF had as well. We bought it jointly with to save money. We don't have a lot of compounds. So one of the things about that is everything's then done with pin tools, so we only transfer 100 nanoliters at a time. No, we never had any powder. Bought it as liquid. No, which is why you can cherry pick once. So you want to do all your assay, all those counter screens, you want to do them all at the same, all the ones that you're going to do at the initial hits, you want to do them at the same time. And then, you can do dose response on them. Then you're going to have to order powder. Sorry. What?
Are you able to estimate how?
Well, that's the only thing I know about the diversity of the library is the cluster size. So, I mean, do you know? So it's extended connectivity fingerprints 4 with a 10-nanometer cutoff of 0.8. And in that, there are no clusters bigger than 2, I don't think, and there might be a few. I take it back, but they're very few. And what?
I'll show the one cluster.
Oh, okay. Yeah. No, but those actually violate the 0.8, Tanimoto 0.8, yeah, because they have too much the stuff on the app. And yeah, about 60% singles, and most of the rest is 2. So, and I'll tell you why. So there are a couple of things about that. One, practical. Roche had a tremendous amount of experience with that. They had several 100,000 compound library and mixtures, self-deconvoluting mixtures. What's wrong with it? Solubility. Basically, if you can get something in at 10 micromolar, it turns out you can't put another thing in there at 10 micromolar too. It's basically having 2 organic compounds in there. Now you can only have them in 5, and it just goes downhill from there. So, yeah, what they found is in the self-deconvolutioning mixtures, very often only one of the 2 hit, even though you should get hits in 2. Doesn't work well.
Compounds?
Yeah. They're all commercial compounds, so they're all in PubChem.
But how many?
Oh, only a couple thousand are drugs. Oh, PubChem isn't a drug database; it's a compound database. It has these days 16 million, something like that. Oh, okay. Yeah, so unless you're screening drugs, the final thing that you're going to put into humans is not going to be what you find in the library. You're going to find something that's 1 micromolar. When he gets done with it, it'll be 10 nanomolar, and it won't be the same structure. And so that's patentable.
Yeah, so then what's the disadvantage? Oh, it's calling PubChem.
Oh, just because if you, I mean, for Stanford, actually not so bad because we work on a lot of things that, I think my old site head's not here. I used to kid him that we work on things here that would get me thrown out of his office. But if you do work on something that pharma's working on, they have all those compounds in their library too. So that's the advantage. If you have unique chemistry, you can be testing. Even if the target's the same, you can be testing new things. In our case, we can only test new targets. The compounds are all plain vanilla.
They don't have the
Right. That's well, and we have a much higher risk tolerance than they do. We do things a lot cheaper and have a higher risk tolerance. There is. Yes. Absolutely. I don't know about frequently, but it does happen. It depends a lot on the assay. So if it's a protein, it's really easy to do that usually. If it's the kind of assay I was talking about that's very biologically relevant, often they're very tricky. And so it's going to be hard. It's going to be hard for somebody else to get it. If the NIH runs it, they want to run it on their fancy robotic system. You may not be able to do that. Yes. Yeah, it would have to be having been a farmer person, it would have to be an extremely cool assay to do that. That's far too early for pharma to give you very much of anything. What? No. I mean, it actually tends to work more the other way. You publish something when I was in pharma, and it was something we were interested in. We would go out and say, hey, can we collaborate? And we'd import the assay. And hard to sell if it's not something they're already working on, because it's too early and they have no idea. They don't know what you know. Well, so.
Okay, now people can hear me. So, and can I use that one?
Yeah,
Thanks. So, so yeah, Bruce told you about high throughput screening. I want to tell you about a complementary approach, fragment-based lead discovery. But I also want to touch on a few points in more detail that Bruce started talking about, which is basically, how do I can...
Oh, it's not auto, sorry.
It's not auto?
I was just pushing.
Oh, you were just pushing, sorry, okay. Okay, so I also want to tell you a little bit about some of the things to avoid. And this applies both to fragment-based screening as well as to high-throughput screening. So in terms of why fragments are of interest, I think that's nicely illustrated by this slide. More than 30 drugs have gone into the clinic, starting from fragments. Some of these are pretty challenging targets too. Venetoclax targets a protein-protein interaction. 2 drugs have actually been approved so far. So this is clearly an area that's been a very rich source of drugs, and it's fairly recent. So 30 drugs in the past couple of decades is quite reasonable, I think. It's something that people are working on in both academia and industry. In blue, we have people in academia working on fragment-based approaches, and in green in industry. So I think there's a lot to offer from both sides. So this, and there's been a lot written on this topic, including 8 books. Kevin mentioned one that I just co-edited recently with Wolfgang Janke from Novartis. And these, I confess I actually do have all of these. They take up a nice little space on my bookshelf. And there's been a lot in the peer-reviewed literature as well. This is just looking at fragment-based drug discovery or fragment-based lead discovery in SciFinder over the years. You can see from the early 2000s, very few mentions up to a pretty nice steady state up here. In terms of how this works, you can think of trying to find a drug kind of like trying to guess a word. And so in the high throughput screening approach, you would take all different possible combinations of letters and screen these to try to find a ligand. In the case of fragment screening, you start not with full words, but with word fragments. You find a lig and an and then link those together. Or more commonly, you would find one lig and then expand that into a ligand. And the reason that these approaches down here are potentially more efficient than the top approach comes down to a question of numbers. There are exactly 101 two-letter words that are allowable in the English, the American English version of Scrabble. And those of you who are hardcore Scrabble players have probably memorized all of these. But if you go from 2 letters up to 3 letters, the number increases to over 1,000, and probably fewer of you have committed all of these to memory. So the same concepts apply to screening molecules. In the case of high throughput screening, you start with a large collection of molecules, find one that sort of binds to your target, and then optimize that. Whereas in the case of fragments, you start with a much smaller collection of fragments, which are much smaller, find a couple that bind close to one another, link them together, and optimize them. Or, more commonly, you would find a single fragment that can then be grown and ultimately optimized. And here, the question of numbers is even more dramatic than for words. There's a famous estimate that the number of possible small drug-like molecules is around 10 to the 63rd. This is a truly astronomical number. It's more than the number of stars in the universe. There's another estimate that all of the compounds in every screening library in academia and industry worldwide is about 100 million compounds. So a planetary-wide screen would not even be covering, scratching the surface of drug space. More recently, there's been a lot of attention on these DNA-encoded libraries, and some of those can be multiple billions of compounds, but even 10 to the 11th really doesn't come close to possible diversity space. So really, it's impossible to fully screen diversity space with any sort of efficiency if you're looking for drug-sized molecules. But as you get to smaller and smaller molecules, the number of possibilities decreases. And Jean-Louis Raymond and his group at the University of Bern has actually been computationally enumerating all possible small molecules having various numbers of atoms. They started with molecules up to 11 non-hydrogen or heavy atoms, and there's only about 100 million of these, of which a measurable fraction have actually been reported as having been made at some point. They then went up to 13 atoms, and the number increases to close to 1 billion. And then more recently, they've gone up to 17 atoms and get up to a couple hundred billion, although they had to make a lot of shortcuts to get to this library. This was actually starting to strain their computational resources. So about half of the molecules that have 17 atoms that have been reported at some point as having been made don't show up in this database. So this is definitely an underestimate, but the point is, as you add atoms, the number of possibilities grows exponentially or possibly more than exponentially. So if you can screen smaller fragments sort of in this space, you can cover more diversity space. There's a few other really nice advantages of screening fragments. One is this concept of molecular complexity, and that is illustrated by this green molecule here, which is perfectly complementary to the target protein, with the exception of this one little nub. And that nub will interfere with binding, and so you won't pick this up at all. Fragments, because they're smaller, have fewer nubs, and so they're going to bind to more sites on more proteins. And typically the hit rates you get are much higher from a fragment screen than from a high throughput screen. A few other really nice advantages are that if you're dealing with a smaller library, it's not going to cost $50,000 and take a year to screen it. You can sort of pay a lot more attention to each individual member of the library. And I think probably one of the key advantages for many of you is that fragments can tackle new classes of targets. High throughput screening collections typically consist of molecules that were made for previous programs. And if you've got some new genomic target that there's really no precedent out there, there might not be anything that's ever been made that could bind to this. But because fragments are so small and so protean, if there is a druggable site on that protein, you'll be able to find it with fragments. Now, another really nice feature of fragments is that even though fragments themselves might be quite weak in affinity, if you can link them together, the binding energies are additive, which means that the binding affinities are multiplicative. And so you can, in theory, take a couple millimolar binders, link them together, and get to a micromolar binder. And by doing so, you can then cover diversity space. So if you have a library of, say, 10,000 fragments that you're looking to bind to two sites on a protein, that's essentially the equivalent of a library of 10,000 fragments linked to 10,000 fragments, or 100 million molecules, basically the size of the worldwide screening collection with just 10,000 fragments. And in fact, this concept was actually first reported almost 35, 36, 37 years ago by the late William Jenks at Brandeis, who described this thought experiment with a hypothetical molecule, AB, with its component parts, A and B, each with their intrinsic binding energies, as well as this connection energy that can be either positive or negative, depending on whether those fragments are productively linked or not. So this was published in 1981. In the mid-90s, researchers at Abbott really reduced this to practice using a technique called SR by NMR, in which they used NMR to find two fragments that can bind simultaneously and then link these together. And this paper really, I think, launched the whole field of fragment-based drug discovery. A lot of companies got started. People started setting up fragment screening efforts in large pharma, academia, leading to the approval ultimately of vemurafenib, the first fragment-based drug approved from a local company, Plexcon, and then more recently, venetoclax, which targets protein-protein interaction. So I think it's fair to say that the past couple decades since SR by NMR have been incredibly productive, and it's been a really rich period for fragment-based approaches. But I think it's also worth asking, why did it take so long to go from Jenks to SR by NMR? And I think initially there was the problem of finding fragments. The challenge is that because fragments are so small, they generally will have low affinity, and so you need to screen at a much higher concentration than you're doing a high throughput screen at. And when you screen at high concentrations, there's all kinds of really awful things that can happen. And if you're not aware of these, they can really sort of derail your screens. And some of these, I'm going to talk about some of the issues that can occur both in fragment screening as well as in high throughput screening. Some of these are actually covered in a review that I co-authored with Ben Davis a few years back. This is actually kind of a fun review. It's open access, so you can get to it even if you're not on Stanford. And what we did is we didn't just go through the published literature. We talked to a lot of friends and colleagues and people we knew and just sort of asked, okay, what are your horror stories of artifacts that you uncovered? So there's a lot of unpublished work in this. It's kind of some things to try to avoid. I think some of these can fall into various categories. Reactive molecules is definitely an issue. These aren't always obvious, but I think there's also the issue of minor impurities, and this is particularly a problem with fragment screening. If you're doing a high throughput screen and you've got some reactive impurity in your collection, in your sample, and you're doing a high throughput screen at 1 micromolar concentration, if you have, say, a 1% impurity that's going to be present at 10 nanomolar, it's probably not going to be an issue. But if you're doing a fragment screen at 1 millimolar concentration, a 1% impurity would be present at 10 micromolar, and that could really throw off your assay. So definitely the higher the concentration you go, the worse these problems can be. And then finally, there's this issue of aggregators, which Bruce touched on, and I've got a few slides on those as well. Now, I think one point that is really important to remember, particularly since many of you are coming with very challenging targets, is that the number of false positives might be relatively the same, but the harder the target, the fewer the hits, and that has really serious consequences. So let's say you have a 1% rate of false positives. That's actually probably pretty low. What would you say, Bruce, is sort of in a high throughput screen? It's 1%. But if your true hit rate is 2%, then that's pretty good. You've got 2 to 1 true hits to false positives. If you've got maybe a little bit more challenging target, your false positive rate might be the same, but now your true hit rate is 0.5%. Then basically 2/3 of your hits are false positives. And if you've got a really tough target where there's hardly anything in your library, basically 95% of your hits are going to be false positives. And this is the thing that's really tough, because you've just spent $50,000 in a year to run the screen. You've got a bunch of hits in there, but you have to appreciate that maybe most of them are not progressible. So you have to be very cautious and assume that everything is a false positive until proven otherwise. So in terms of some of the things that can go wrong, this is a really nice paper that just came out last year from Alessio Ciulli and colleagues at the University of Dundee. They actually did a fragment screen where they were looking for inhibitors of a ubiquitin ligase. And they actually found this fragment that was pretty appealing. It passed a number of biophysical screens. It was actually showed very nice activity by isothermal titration calorimetry. This is gold standard assay. This is the kind of sort of data that you dream of when you do this sort of assay. It was pretty potent, about 15 micromolar, pretty potent for a fragment. And they actually were able to do two-dimensional protein-detected NMR. This is, again, gold standard here. What you're looking at is a few residues are shifting position, telling you that a few residues in the protein are being affected by binding to something. And they actually were able to map these on the known structure of the protein. And what they found is that all of these residues cluster right around the active site. Again, just beautiful data. You'd be hard-pressed to dream of something better than this. The problem was when they started doing medicinal chemistry on this, they just couldn't get anywhere. Most of the compounds they made were inactive. A couple of them were active, but they really didn't make sense why some were active and some weren't. They went back and remade this, and it turned out actually it was not active. And they were ultimately able to get a crystal structure. And what they found is that the activity in the NMR assay and the isothermal titration calorimetry assay was not coming from their fragment. It was actually coming from what they determined was zinc, this metal ion, that they could identify crystallographically as actually binding to the active site cysteine and active site histidine here. So this, I think, illustrates the point that they did everything right. And they actually went back and looked at the NMR and the mass spec of this. It looked great. It's really hard to detect something like zinc. And so the first thing when you get a hit, whether from a fragment screen or a high throughput screen, is you always want to go back and get that compound from another source and retest it and make sure it confirms. So this is I think, something that everyone can agree is false positive. The next topic is a little bit controversial, and so I want to preface it with a quote by the RAF pilot and escape artist Harry Day. Rules are for the obedience of fools and the guidance of wise people. What I'm going to talk about is something that generates quite a bit of debate among chemists, and since all of you are wise people, I think take what I'm going to say with guidance and not as sort of hard and fast rules. And it turns out that a lot of small molecules can act through mechanisms that are not particularly drug-like. This is an example. It's called rhodanine. This actually is can act covalently. It's also can be fluorescent, and it's also photoactive. And so these types of molecules come up as hits in a lot of different assays. People file patents on them. People publish on them. Computational chemists see them coming up, and they think that they're privileged pharmacophores. And Jonathan Bale, a few years back, came up with this concept of pan-assay interference compounds, or PAINs, that he and a lot of others feel are polluting the scientific literature. And what people have been doing is going through and trying to systematically identify some of these pharmacophores as things to watch out for. Now, the reason I say this is controversial is because before Steve says it, I will say that some of these substructures do appear in approved drugs. And so it's not necessarily the case that a PAINs molecule is a priori acting pathologically. But if you go back to that notion of sort of false positives versus true positives, the odds are sort of raised against you if you have a molecule that has one of these substructures in it. And Mike Walters and Jonathan Bale wrote a really nice comment in Nature a few years back, where they came up with this notion of PAINs as chemical con artists. All of these look like genuine hits, and they might actually even exhibit some SAR, but you can imagine if you follow any of these characters for very long, they're probably going to lead you to a fairly unpleasant place, and you might want to avoid them. And I think the easiest way of avoiding them is just not putting them in your library in the first place. This is actually from a pretty high-profile research group in a pretty high-profile journal, and it's a fragment library that, when you look at it, it's just lousy with PAINs. They've got catechols and quinones. They liked that one so much they included it twice. And it also has this molecule here, toxaflavin, and I just wanted to take a few slides to show you how pernicious these types of molecules can be. So toxaflavin is a natural product. Back in 2003, researchers at Roche published a really nice paper where they found that toxaflavin can inhibit a protein tyrosine phosphatase, PTP1B. This is an enzyme that has an active site cysteine that's very sensitive to oxidation. And what they found is that toxaflavin, in the presence of common reducing agents such as DTT, can be reduced and then spontaneously oxidizes in the presence of molecular oxygen to generate hydrogen peroxide. And hydrogen peroxide can then go on to oxidize the active site of PTP1B. Hydrogen peroxide, of course, is also a second messenger. It's a very potent cell-based signal. So this paper was published in 2003. A few years later, researchers at GlaxoSmithKline described an assay for detecting this type of what they call redox activity, the ability to generate reactive oxygen species in the presence of common buffers. And so this came out in 2007, and they showed various molecules that can exhibit this effect. But nonetheless, in 2008, we had a pretty high-profile academic publish a paper on a novel class of small molecule inhibitors of HSP90. When you read the paper, it's very clear that they weren't aware that these are acting through a redox mechanism. Shortly thereafter, we had a paper targeting an antibacterial target. Then we had another paper looking at toxaflavin analogs inhibiting this very interesting protein-protein interaction. I think they could retitle this paper Investigation of Hydrogen Peroxide as a small molecule antagonist of this interaction. There's, CERT1 was a big target a few years back, and of course toxaflavin or hydrogen peroxide inhibits that. One of my favorites is this paper. This was actually, they did computational screening and identified toxaflavin analogs as inhibitors of a different protein tyrosine phosphatase. Again, a protein that has an active site cysteine, and they actually went so far as to misspell the pharmacophore here and suggest that it acts as a phosphotyrosine mimetic. So this came out 10 years after that initial paper from Roche, but no one had bothered to look at the literature and check to see if these are actually acting through a pathological mechanism. Also in 2013, we had another paper that kind of starts ominously. Toxaflavins as selective small molecule inhibitors of this enzyme here. But it became clear as you read the paper that they did figure out what was going on. And they actually go on to say, given the prevalence of these compounds in screening libraries, we feel it's important to raise awareness in the community and highlight these as assay interference compounds. We hope this report will alert others to the issue we have experienced, despite apparently clear and rational SAR against our chosen target. So this is 2013. Did they actually alert others to the issue? Well, unfortunately, no. This is from 2018. Again, a very close analog of toxaflavin, coming out of a screening assay. This is something, it's not just toxaflavin. There's a lot of molecules that can exhibit this sort of redox activity. These appear in both the NIH and LOPAC collections. There are ways to detect this. You can actually add catalase to break down the hydrogen peroxide. Switch to a more modest reducing agent sometimes helps. You can actually directly detect hydrogen peroxide production. And I think talking to a chemist is probably just a generally good idea. And this is something that it's not just in NIH collections. And unfortunately, these also appear in Stanford library as well. These came out of a current Spark project as hits against a target. Fortunately, in the MedChem working group, they brought these to us and they were like, oh, we're having trouble finding commercial suppliers of these, which I was actually happy to hear. But what was interesting about this, though, is actually apparently toxaflavin is in the assay, and it didn't come up as a hit. So you actually can get SAR even with pathological mechanisms. People sometimes refer.
About half of the things that have that symptom
came up as hits.
were hits.
Yeah. So I mean, it's it's people sometimes refer to. Instead of structure-activity relations, structure-interference relations as sort of the... What is nice, though, about pains is that you can identify them. And talking to chemists, Steve and I might give you sort of different opinions as far as like how far away to run. But we'll generally be able to recognize them and say, okay, be cautious. What is sometimes, what I like to think about pains compounds is, I'm reminded by this, of this cartoon from XKCD. When you see a claim that a common drug kills cancer cells in a petri dish, keep in mind, so does a handgun. I think pains are kind of the handguns of the chemical world. Sure, they're probably going to hit your assay, but it's not a very subtle tool for probing biology. But as I said, at least you can identify those. There's another problem that Bruce touched on that is in some ways even more insidious, and this is the fact that a lot of small molecules will form aggregates in aqueous solution. These can actually be visualized using electron microscopy. And these aggregates will just non-specifically interfere with a lot of different types of assays, whether those are biochemical assays or cell-based assays. And this is something that happens not just with really ugly molecules, such as tetraiodophenylphthalein. Even approved drugs can form aggregates. All of these are approved drugs. They all have specific targets at a therapeutically relevant concentration. But at higher concentrations, these will form these aggregates that will just non-specifically inhibit a variety of different assays. So how much of a problem is this? This is kind of a chilling paper that came out. There's actually a Stanford postdoc who had done his graduate work with Brian Scheuchert, who worked on this paper, where they were interested in AMP-C beta-lactamase. They did a high-throughput screen. Unlike the high-throughput screens here, this was done full dose response curves on every single compound, 70,000 molecules, 4 nanomolar to 30 micromolar. And they actually got a pretty decent hit rate, almost 1,300 hits. Of those, 95% of them were sensitive to detergent. Remember, Bruce said, So remember, Bruce said, always screen with detergent. That initial screen, actually, they didn't include detergent. And detergent is the easiest way to weed out aggregators. If you can't screen in detergent for whatever reason, when you follow up on your assay, if you add detergent and your activity disappears, that's almost a sure sign that you'll get an aggregation. Of the remaining 70 hits, 25 were beta-lactams. It's a beta-lactamase, so no surprise there. 24 just didn't reproduce at all. 9 were also aggregators, but had a little bit more sort of challenge identifying them. And a dozen were covalent inhibitors. There was not a single true non-covalent hit out of this entire screen of 70,000 molecules. And so I think this is just very important to remember that you might not get any hits at all out of your library, or let me rephrase that, you might not get any real hits out of your library. And I think every time you go into a screen, you have to be prepared for that possibility. Unfortunately, not everyone recognizes this, and so you actually get aggregators in the published literature. There's an anti-cancer target called lactate dehydrogenase A, LDHA. People have been trying to find inhibitors of this for some time. There's a fairly unattractive molecule with a rhodonine and a catechol that came out a few years back. More recently, there was a slightly less unattractive molecule, modest activity, 8 micromolar, still a catechol here. But when researchers at Ariad wanted to start a drug program on this target, the first thing they did is they went back and they made and tested some of the compounds that had been reported in the literature, including these two. And what they found was that in the presence of detergent, no enzyme inhibition was observed for these compounds when tested up to 1 millimolar. And just to kind of twist the knife, they had, not surprisingly, attempts to produce co-crystal structures were unsuccessful. So I think what this says, basically, is that these papers are garbage. And that's kind of embarrassing for the people who publish them, but it's also kind of scary because when you look at the number of citations, almost 500 citations of this paper since 2010, and this is actually sold by a number of vendors as a chemical probe for LDHA. So it's not just looking at your own hits that you want to be cautious and assume that everything is an artifact until proven otherwise. When you look in the literature, you also want to be very, very cautious about the types of things that you find there and just make sure that they've been validated or validate them yourself. Please. Right, so the question is, if you screen in the presence of detergent, will that affect the binding between the molecules in the target? And yes, that can be an issue. Some targets are sensitive to detergent. Oftentimes, people can use different detergents if you have a particularly sensitive target. There's a bunch of non-ionic detergents, and people will sort of go through all of them. Does anyone, I mean, are there assays where, cell-based assays, detergent can be a challenge. So yeah.
Below the CMC.
Yeah, exactly. So yes, it can be an issue, and so that's where if you do get a hit, I mean, I think coming back to what Bruce said early on, really try to optimize your assay, and if at all possible, include detergent, even if that takes a few extra weeks to try to work out. If there's no way you can do that, then you just have to be extremely cautious with any hits you get and follow very closely. Yeah.
What are the ways
Yes. Yeah. So some of the So Bruce makes a very excellent point. Orthogonal assays are great. So SPR, for example, can show these sort of aggregates will produce these very, very high signals, for example. Yeah. Yeah, so the question is, does adding more DMSO solve the problem? Not really, because even if you can go up to 10%, it's still 90% water. And I mean, the thing with aggregates is different proteins have different sensitivity to aggregates, so it's not even the kind of thing where you can screen out your library. You can have molecules that aggregate and may interfere with a lot of different proteins, but under other conditions they might not aggregate and might be true hits. So it's really something that you have to look at on an individual basis. I mean, all of this sort of, I think, illustrates why it took so long for fragment screening to catch on. If you screen at high concentrations and you find aggregators, for example, this whole phenomenon of aggregation really was not even well understood until the early 2000s. And it's really sort of took a while for people to figure out how to get around this. And it does raise the question, how do you find fragments? And there's actually a lot of different methods. Bruce touched on some of these. I think biophysics are really key because they can show you direct binding oftentimes between your molecule and your target. And this applies not just to fragments, but high throughput screening hits as well. If you can use biophysics to sort of confirm binding, that's very powerful. But again, as Bruce said, I think you want to use combinations of these. Every screening technology has its false positives and false negatives. And strengths and weaknesses. And so combining several, I think, can be really important for distinguishing true hits from false positives. And I think this is what people are doing. This actually looks at a few polls that I've done on practical fragments, starting in 2011, asking people what screening technologies they're using. And what's interesting is this has gone from the average respondent using just over 2 techniques to the average respondent using 4 techniques in 2016. People really are using multiple different orthogonal assays to confirm hits, with NMR and SPR being the sort of the leaders. But what's interesting here is X-ray crystallography. Look at how much this has increased from 2011 up until 2016. Crystallography is really catching on, and I think will continue to expand. So just a couple of definitions as far as what a fragment is. I think many of you are familiar with Lipinski's rule of 5. When the concept of fragment-based drug discovery was first being codified, researchers at Aztecs came up with the rule of 3, sort of a stripped-down rule of 5. In reality, people are moving to smaller and smaller fragments, and the reason why, I think, is shown nicely here. This is also from Aztecs, and what they did was they looked at the hits that came out of their screens that they could confirm crystallographically, sort of gold standard, and they looked at the properties of those hits compared to the representation in the screening library. So if you look at this column out here, about 12% of their screening library consisted of fragments with more than 17 heavy atoms, but only about 6% of their hits had more than 17 heavy atoms. Whereas here, about 11% of their screening library consisted of fragments with 12 atoms, but these were overrepresented among the hits. And this is, I think, just a really beautiful illustration of that molecular complexity hypothesis. As you get larger, there's more little nubs that will keep these things from binding. And so there is sort of the sweet spot in sort of the 8 to 14 atom range. Now, Bruce touched on the Stanford fragment collection. It's around 5,000 molecules, and this is really set up for SPR screening. I don't know if there's anything you want to comment on. Yes, which is exactly, yeah. So, I mean, I think this is, you know, if you are interested in fragment screening, definitely talk to Bruce, because I think this can be very powerful. In terms of how to evaluate fragment hits, those of you who are medicinal chemists and come from sort of a more traditional background and are used to seeing the output from high throughput screens, you know, generally you're looking for molecules that are, say, micromolar or better. Fragment hits might be very weak. They might be millimolar, and it can be very hard to convince a medicinal chemist to get excited about that. One way you can sort of put it in perspective is with this notion of ligand efficiency. This is a very simple concept that was first proposed by Andrew Hopkins, and it's simply the binding energy divided by the number of heavy atoms. This was based on some work by Tak Kuntz up at UCSF, where they had plotted about 140 protein-ligand interactions against looking at the number of heavy atoms versus the affinity. And you get this very steep slope initially, but then it starts to sort of level off, and down to about 20 atoms or so, each additional heavy atom you add is not that great. In general, what you're looking for is a ligand efficiency in sort of the range of about 0.3 kcals per mole per atom. That is a number that means that your final molecule would have a molecular weight of around 500 and 10 nanomolar affinity. So above 0.3 kcals per mole per atom, that's even better. Below that, it's just going to be a lot harder to get something at the end of the day that has both affinity and small size. Okay, so those are some of the concepts and some of the things that can go wrong. I want to close with some of the things that can go right and talk about a few success stories out of fragment screening. The first is, I think, just really nicely illustrates this notion of fragment growing. This comes from Aztecs. They were looking for inhibitors of HSP90, anti-cancer target. They did a screen of a pretty small library, about 1,600 fragments. They got about 125 hits, so you can get a very, very high hit rate here. They put all of those into crystallography and got 24 structures. This actually was not the best hit you know, it's pretty weak, close to a millimolar binder. Ligand efficiency is really nothing to write home of. But what they noticed when they looked at the crystal structure is that methoxy group is in a lipophilic region of the protein. And so just by replacing that methoxy with an isopropyl group, they could gain 2 orders of magnitude affinity. They also recognized that this portion of the molecule is pointing to another hydrophobic region of the protein. So by building out here, they could gain another 2 orders of magnitude affinity. And then looking at some of the other molecules that have been published in the literature, they added a hydroxyl over here and got yet another 2 orders of magnitude in potency. So just by adding 6 atoms, they've improved the affinity by about 1 million-fold. This molecule actually had lousy pharmacokinetics, and they couldn't get rid of these hydroxyl groups. They're really essential for binding. But what they were able to do was to change the overall physicochemical properties of the molecule by adding this piperazine, and that then gave them a molecule that had sufficient pharmacokinetics to go into clinical trials. What I like about this example, in addition to just the really creative use of structure-based design, is when you look at the final molecule in gray compared to the initial molecule in blue, you can see that they're really occupying the same space. And this is something that you often see in fragment-based drug discovery, that the fragment is really making some of the key interactions, and they're just sort of adding new interactions here and here and here, but the core fragment is maintaining the same position and the same orientation. This is common, but as I'll show you in a couple slides, it's not universal. Now, the first approved drug to come out of fragment screening started with a fragment screen against kinases in general. They were actually looking for just sort of generic kinase, what are called hinge binders that bind in the ATP binding site. They got this azaindole, which is very weak. It binds in multiple orientations and multiple kinases. But they were ultimately able to optimize this to a highly selective molecule that inhibits a specific form of BRAF. And this was approved as Zelboraf for metastatic melanoma a few years back. And I think this illustrates that you can start with a very non-selective fragment to get to a selective inhibitor at the end of the day. And just sort of driving that point home, this is azaindole leading to vemurafenib. Plexicon also started with 7-azaindole to come up with a different series of compounds that hit a different kinase, CSF1R. And independently, researchers at Aztecs and AstraZeneca also started with 7-azaindole to come up with an inhibitor of the kinase AKT. Same starting point, 3 totally separate molecules against 3 separate kinases, all with defensible intellectual property. And I think this illustrates a very important point, which is that you don't necessarily need selectivity for a fragment. So, you know, Bruce just mentioned if you get the same compound is hitting in a bunch of different assays for high throughput screening hit, that is potentially a problem. But with a fragment hit, again, because they're so small and you need to build them, that's not necessarily a problem. If your fragments were too selective, you wouldn't be able to get hits out of a 5,000 fragment library. Now remember I told you that the compounds often will maintain their position and orientation as you optimize them. This is a counterexample. This is another program against HSP90, this time from Nerviano. They did a fragment screen, found this kind of interesting molecule, and looking at what was also reported in the literature, they recognized that resorcinol fragment here shows up in a lot of HSP90 inhibitors. And so they just added this on to this. They got a modest improvement in potency. They actually got a crystal structure, found that it binds in the design fashion here. But what they also did is something that is extremely wise, which is that they screened a synthetic intermediate shown here. And this was actually more active than their design compound. And it's kind of baffling when you look at the structure. There's no way these methyl groups could fit back here. And actually that turned out to be the case when they solved the crystal structure of this molecule. They found that it's completely flipped over. So this is, I think, illustrates the fact that even though fragments often maintain the same position and orientation, that's not universal, and you always have to be prepared for surprises. And again, crystallography can really help you there. They were ultimately able to take this down to a low nanomolar inhibitor nonetheless, but this would have been a lot harder to do had they not recognized this different binding mode. So these are examples of fragment growing, which I think is probably the most commonly used approach. But there is something really seductive about this notion of finding two really weak binders and linking them together. And in fact, one of the very first published examples of fragment screening to come out of Abbott was actually that very process. They were looking for inhibitors of mp4. They found in the presence of this hydroxamic acid, which binds to the catalytic zinc, they found this biphenyl that they could then link together to get a nice 25 nanomolar inhibitor, which after some pretty heroic medicinal chemistry could get to a drug that went into the clinic. But what's interesting about this, when you look at the details and you look at the SAR around the linker, a 2-atom linker gives you a 25 nanomolar binder. 1 atom shorter, you lose an order of magnitude binding. 1 atom longer, you lose 2 orders of magnitude in binding. So you have to be exactly right if you're going to get this to work. If you're off by a fraction of an angstrom, all of the potential benefits you might get by linking are going to be lost. That said, this can be very powerful. The second drug approved out of fragment-based screening also came out of Abbott. And in this case, they were looking for inhibitors of protein-protein interaction. They found these very weak binders, 300 micromolar and 4 to 6 millimolar. They could link these together to get to low micromolar binders. They reached sort of this affinity plateau. They realized this phenylalanine was keeping these from binding in the lipophilic pocket, so they redesigned the molecule, replaced the carboxylic acid with a bioisoster, this acetosulfonamide, and got to mid-nanomolar binders, ultimately getting to a molecule that went into clinic, which they note, actually, this is far from what's traditionally thought of as drug-like and violates 3 of the 4 rules of 5. This inhibits both BCL-XL and BCL2, and they realized they wanted a selective BCL2 inhibitor, so they did further optimization on this, ultimately getting to venetoclax, which has been approved for CLL and is actually quite an effective drug. And I think this is a nice example of breaking rules judiciously. Again, this really is not a particularly drug-like looking molecule. It's even got a nitro group on there, which is something that scares a lot of medicinal chemists, and yet it's a great molecule. So you always want to be you want to know what the rules are, and you want to know why they exist, but you also want to be willing to break them if the data suggests that that's productive. Now, in terms of fragment growing versus fragment linking, as I said, that fragment growing is much more common. This is from a poll I ran a couple years ago. And just asking people's success with fragment growing or fragment linking, where you can see in red, people found that fragment growing generally works pretty well. Fragment linking, interestingly, about 1/3 of the respondents had never tried it, but of those who had, it either worked okay or worked well. So for tough targets, this actually can be very effective. And I think just to give you a sense of how much of a boost in potency you can get, this is a retrospective example where researchers at Sanofi took a known inhibitor of factor Xa, 2 nanomolar, and they just asked what happens conceptually if you break that bond. You end up with a 60 micromolar fragment and another fragment that's so weak that you can't actually detect binding. The affinity you get by linking these together is greater than the affinity of that fragment by itself. So you can get orders of magnitude improvement in affinity if you can get proper linking. So just closing with a couple of slides. Coming back to that slide I showed you at the very beginning of the number of drugs that had come out of fragments. If you look at how those break down into target classes in red, and then there's a couple reviews that I've co-authored looking at fragment to lead success stories in 2015 and 2016. Those are shown in blue. You can see it's a pretty wide range of targets, kinases, proteases, enzymes are all well represented, but protein-protein interactions are also fairly well represented, and even more baroque targets such as transcription factors and a GPCR in here. So I think there's really a lot of opportunity for fragment screening against a variety of different targets. Just a few resources to leave you with. I've mentioned a couple times this blog I run, Practical Fragments. There's a LinkedIn group as well. I didn't have a chance to tell you what we're doing at Karma, but what we really focus on is not finding the initial fragment, but sort of that second step of how you advance a fragment to a more potent molecule. And there's also a review that I co-authored a couple years ago that has a lot of this material in it. And then finally, if you're interested in this to speak, there's some nice events coming up, both in San Diego and in Boston later this year. Check these out, and just like to thank Kevin for inviting me and Bruce for co-speaking, and all of you for sticking around. Thanks. And happy to take questions. Yes.
Software that you know take two fragments and calculate various factors that might work?
Yeah. So the question is there software that can take two fragments and then link those? And there actually is there's several programs out there that people have tried to do this with. One that you can actually get free, that you can actually try for free, is called Biosolve IT. And that actually seems like a fairly useful software tool. I haven't actually used it myself, but it seems fairly impressive. There's another one that was actually just published recently that starts with a fragment and then sort of tries to dock a second fragment nearby, and that is from an academic lab, and all of that is open source. So yeah, there's a number of programs that people have written for that.
Something like that might be
Yeah.
if you have a say, how else could I connect these?
Yeah, yeah.
Creating two fragments from
Right.
a line.
No, that's you know, and so the comment is this could be useful for scaffold hopping, and I think actually that's one thing I didn't get a chance to talk about. I think fragment screening and high throughput screening can be very complementary, because oftentimes what people have done is they'll do a fragment screen and they'll get hits, and then they'll look at where those hits are binding if they have access to structural information, and they might be able to map those to the location where one element of a high throughput screening hit is binding, and that can sort of guide them into new chemical space. They might have there have been cases where, say, a high throughput screening hit has some tox problems, and so doing a fragment screen, they can swap out that element with something they get from a fragment screen.
Yeah, thank you.
Oh,
That was-
sure.
That was really terrific.
Thanks.
Really amazing.
Oh, no, thanks.
Very cool. In terms of making slides available, so you've sent... I've already sent.
Yeah, I'll send, what I'll do is I'll send a PDF, if that's okay?
Yeah, no, that'd be fantastic.
Yeah, no, I can do that. Yeah, I hope I didn't, I hope I didn't go too long.
Yeah,
Hey,
pleasure.
good to see you.
Congratulations.
Thank you.
Getting money.
Yeah. No, it's,
Long run, actually.
yeah, no, it's,
Around 8 years.
well, you know, the old saying is 90% of success is just showing up. In biotech, 90% of success is just staying alive. It's surviving. So yeah, we've
happened so many times.
Yeah. No, so we've
gone under.
Exactly. Yeah.
That's true. Douglas company,
Exactly. Was Sun. Yeah. Exactly. And
It's true.
then a
Drug
pretty
discovery
nice.
programs in big companies too.
And also.
Many of them have had experiences. Oh, from 91 till Roche bought them, and then I was at Roche until they closed Roche Palo Alto.